The purpose of this research is to investigate ways to slow the coral bleaching process and ultimately lower coral mortality rates during and after a bleaching event. On the left here, you can see a healthy elkhorn coral, and on the right, a bleached elkhorn coral. Coral bleaching happens when ocean temperatures rise and coral expel their algal symbiont, Symbiodinium. Coral polyp tissue is mostly transparent and colorless, so when they lose their algal symbiont, you can see right through it to the calcium carbonate skeleton underneath, giving it that bleached appearance. At this point, the coral isn't dead, but because it relies on its symbiont for over 90% of its nutrition, unless it gets its symbiont back quickly, it won't have enough energy to fight off disease, and it will eventually starve. There are a number of things that can cause coral to lose its symbiont, but the most common, widespread, and rapid cause of bleaching is heat stress. On the bottom left, you see a cross-section of a polyp. Above that is a zoomed-in view of the cells lining the stomach cavity. This is where the algal symbiont, Symbiodinium, is located. It's surrounded by a membrane that makes up the symbiosome. In the top right corner, we zoom in further to see the thylakoid membrane where photosynthesis takes place inside the Symbiodinium. When temperatures rise, the thylakoid membrane begins to destabilize, disrupting the electron transport chain. As unpaired electrons flood the system, they react with oxygen and form a variety of toxic reactive oxygen species, or ROS. There are several different methods organisms can use to deal with reactive oxygen species. The mechanism I focused on is the superoxide dismutase enzyme, which converts the highly toxic radical superoxide anion into less toxic hydrogen peroxide. I also used Aiptasia, which is a common model organism for coral research. Because ROS primarily form in the Symbiodinium, where photosynthesis takes place, I wanted to isolate bacteria located in and around the symbiosome. To do this, I macerated Aiptasia polyps and centrifuged the tissue in a discontinuous density gradient. The first image on the left shows a test tube with a discontinuous density gradient and macerated Aiptasia tissue floating on top before centrifugation. The second image shows the density gradient after centrifugation. You'll notice there's a dark mass floating lower in the test tube. That's because the Symbiodinium cells have a higher density than the host cells, and they settle at a higher density gradient. Next is the same density gradient with a little bit of food dye and no Aiptasia tissue. This was just to confirm that the density gradient stayed intact during centrifugation. The center image, top left, shows the Symbiodinium cells immediately after separation. They are the perfectly round green and brown cells you can see there. You can also see there's still a lot of other stuff mixed in with the Symbiodinium. To clean these cells up and increase my chances of getting the bacteria I want, I filtered these cells through a series of sieves with increasingly smaller holes. Then collected the Symbiodinium cells, resuspended them in sterile artificial seawater, and I did a serial dilution and plated the Symbiodinium cells. The final image shows the colonies that grew, and there are arrows indicating the morphology of the colonies that were selected for this study. Once I was able to establish a pure culture of this bacteria, I identified it using the 16S gene. The next step was to find the superoxide dismutase gene, or SOD gene, in this bacteria. In order to do this, I generated a phylogeny in the NCBI database from the top 13 closest matches I got when I ran a search on the 16S gene. Then I looked up the SOD gene for as many species and strains as I could find from the more closely related subset of species highlighted by the yellow box. Then I created an alignment of the SOD genes I found and used the conserved regions to design primers. You can see the results from the gel electrophoresis in the blue box on the right. The arrow is pointing to the bright band of amplified SOD gene DNA. Once I had the SOD gene sequence, I used Exaiptasia to find the open reading frame and convert the SOD gene sequence into its amino acid sequence, which is that string of letters at the top. From that, I was able to identify the type of SOD gene based on two key features of this amino acid sequence. First, the underlined sequence is highly conserved in the manganese-iron SOD gene family. And second, the relative spacing of the four specific amino acids, which are highlighted in yellow. When the protein is folded, these yellow highlighted amino acids will form the active binding site for the metal ion that stabilizes and catalyzes the SOD enzyme. The image on the left is the structure of the folded protein, and the image on the right is that same protein with the active binding site formed by those key amino acids, highlighted in red. Next steps for this research would be to identify bacteria that are able to upregulate SOD enzyme production when it detects signs of photo-oxidative stress, and then use these bacterial species as part of a probiotic mix administered to its host prior to heat stress.